All right, in this video, we are going to discuss the electron transport chain. So the whole point of oxidizing fuels to get those high energy electrons is so we can get them to the electron transport chain so that that energy can be used to create a proton gradient. And that's what we're going to look at in this video. So remember what we've looked at previously. So when we looked at the citric acid cycle right here, we saw that it generated electrons that were carried by NADH and FADH2. We also saw some electrons generated as we went through our glycolysis cycle. And all of these things are going to find their way down to the electron transport chain. And that's what we are going to look at. So the citric acid cycle oxidizes the acetyl fragment of acetyl-CoA to carbon dioxide. So we give off that carbon dioxide. In the process of oxidation, we got high energy electrons that we captured right in the form of NADH and FADH2. And so the key function of that citric acid cycle was to harvest these high energy electrons from the carbon fuels. And those activated electrons are carried by these carriers here. The citric acid cycle occurs in the matrix, the mitochondrial matrix, so in here. Okay, so that's where those high energy electrons are generated. There are mechanisms for NADH that's originating elsewhere to make it into this matrix. And inside this matrix is also where the electron transport chain and oxidative phosphorylation will happen. So the proteins that are going to catalyze all this are on this inner membrane. All right, so let's look at that. And let's kind of orient ourselves here. So all in here, we're in the matrix. You see that's labeled. This is that inner membrane. Okay, here is our intermembrane space. Not shown, but here would be the outer membrane. Okay, and then outside of that would be the cytosol on the other side of that. So for the electron transport chain, we want to think about how energy flows through redox centers, areas of oxidation reduction, that are part of these electron transport chain enzyme complexes. And we want to quantify the energy transferred so that we can understand where the energy comes from to pump protons from the matrix out into that intermembrane space. And you can see that happening several places along here. So today we're going to look at complex 1, which is a portion of this electron transport chain in detail, and then we'll look at the other ones, but in less detail. Okay, so electrons are generated by oxidative reactions they're carried by NADH, FADH2, that ubiquinone that we've talked about just a little bit. And they're passed from reduction center to reduction center and ultimately to oxygen. So see, we've got oxygen here, and that oxygen gets reduced to form water. Okay, so we can see our 4 complexes. 1, 2, 3, 4. Complex 5 is ATP synthase, and that's in our next video. Okay, so as electrons are carried through these complexes, the complexes harness that energy to pump these protons from the matrix into the inner membrane space. And you can, if you look at this in detail, you'll see complex 1 is a pump, complex 2 is not, complex 3 is a pump, complex 4 is a pump. All of those are pushing those protons out. So this is a model, cartoon drawing, and we also want to look at the kind of theoretical mathematical model. And if we can show that the amount of energy gained from dropping these electrons from high energy to lower energy equals the amount of energy needed to pump these protons across that gradient, then that's going to be evidence that we have a good model. All right. So just a few more notes. First of all, respiratory chain, okay, is another word or another term for the electron transport chain. Okay, and so this is a little bit of what we just said, that this respiratory chain catalyzes the flow of electrons from low reduction potential carriers. We're going to review our electrochemistry, to high reduction potential. So essentially, but notice it says exergonic. These are going from higher energy, less stable, to lower energy, more stable. So they're giving off that extra energy. Okay, those electrons are eventually transferred to oxygen. Okay, and the energy is used to establish a proton gradient. And so next we're going to review this relationship, free energy and electric potential. Before we do that, let's look just a minute at that same picture. And I just want to remind you that the name of an enzyme complex gives you a clue to its function, right? So when I look at complex 1, NADH coenzyme Q reductase. It's a lot of words, but think about what that is. We know reductase, we're going to be looking at a transfer of electrons, and this is telling you that electrons are being transferred from NADH to coenzyme Q. Okay, if I look at the name of the second one, complex 2, succinate coenzyme Q reductase. Hey, look, electrons are picked up from the succinate. They're passed to FADH2 and then to ubiquinone, which is our Q. Right, let's write that here, Q. And so just a kind of reminder that the name gives you a feel for what might be happening. All right, let's review our general chemistry a little bit. So this might look familiar. In chemistry, we talked about electrochemical cells, and we talked about the fact that if we set up two half reactions, so this side has just zinc and zinc 2+, and this side has just copper and copper 2+. There's no route for the copper ions or the zinc ions to flow from one to the other. But what we find in this particular case is that the electrons flow spontaneously from the zinc to the copper. Okay, remember that redox is always coupled. If we have oxidation, we have to have reduction. So if electrons are moving, if zinc is giving away electrons, copper must be taking them. If zinc is being oxidized, giving away electrons, copper must be being reduced, taking those electrons. So when we have that flow, we have a potential difference. And anytime we have that, we can do electrical work. And you might remember a chart that looked something like this. There's various versions of it, but our standard reduction table. Higher values, okay, more positive. Standard reduction potential told us that there was a strong driving force for the half reaction to occur in this direction. So all of these half reactions are written as reduction reactions. And remember, none of these are full reactions. They have to go hand in hand with an oxidation reaction. So the driving force for fluorine to pick up electrons to become fluoride ion is really high. Okay, there is a stability associated with it. The electrons really want to be in that fluoride ion. On the other hand, if you come down here to lithium, okay, this has a really negative number. That tells us there is not a strong driving force for this to move in that forward direction. But instead, what's really likely is for it to occur in reverse. Okay, so we would expect that that lithium would be fairly easily oxidized. It would give away its electron because it has this really negative value. So when we pair two reactions, we can look at what we call the potential, right? And potential is that symbol. We can find the potential for the cell for the overall reaction by taking the value of the reduction potential for whatever's being reduced minus the value for whatever is being oxidized. Okay, so for example, for our zinc, copper one we were looking at, zinc was giving up electrons. It was being oxidized. It goes over here. So minus negative 0.76. The copper was being reduced. It goes here. And I get 1.1 volts. It's a positive value. That told us it was galvanic, which meant spontaneous. Okay, you could also have electrolytic cells, non-spontaneous. Okay, so there was the overall thing. We're going to tie this back into the biochemistry, I promise. Okay. So this is where I was going to do that math that I just already did. So let's keep going here. We want to relate that cell potential to the free energy. So the change in Gibbs free energy for an electrical system is equal to the negative of the charge times the voltage being that potential. Okay, so voltage is the cell potential. Charge is the moles of electrons times Faraday's constant. Okay, and so we get at the end this equation. This says delta G is negative NFE. This little degree symbol still means we're in standard state, so 1 molar concentrations. And we know that 1 molar concentrations are actually pretty rare, so we want to be able to talk about non-standard cell potentials. So starting with our delta G reaction, and then plugging in negative NFE every time we see delta G. So delta G, negative NFE. The delta G standard right here, so we're going to do negative NF times the standard cell potential. And then, highlighted in pink here is our correction term, RT natural log of Q. And then to make this a little shorter, we divide everything by NF, and we get this final equation, that the cell potential is equal to the standard cell potential. This one is the one we can calculate from those standard reduction tables. And then minus this whole piece that is dependent on Q. Ooh, an important point here. Q, remember, is that ratio of products over reactants. Maybe a little confusing because we're also using ubiquinone up there. This Q was that ratio of the products over reactants. Okay, here is a list of all your constants that go with that. Okay. So when we're looking at this, we can calculate the energy that can be harnessed from the redox potentials of the electron carriers in that electron transport chain. And then we can adjust from our standard conditions, okay, and we can calculate that energy that we get as we move down the electron transport chains and compare the energy we should get from those electrons moving to the energy needed to pump out the protons and see if it adds up. We will have practice problems on this at the end of the slide deck, okay, on doing these actual calculations. Couple things to note. So it looks a little different, right, when we're looking at our half reactions now. One thing I want to really highlight is that this table has the more negative values up top. So the things that are less likely to occur as reduction reactions are up top. And if you come down here, notice that oxygen reacting with hydrogen to form water is a really favorable reduction reaction. Okay, so this is where the electrons really want to be. This is the dumping point for all those electrons. We can use this to calculate the free energy. So for example, free energy of taking electrons from NADH and adding the electrons to oxygen. Okay? So electrons from NADH and then taking them to oxygen. If I think about that, electrons, taking electrons from, losing electrons, this will be oxidized. And we're giving them to oxygen, so oxygen will be reduced. So when I look at this for my reduction potential, okay, I would come down and find the value for oxygen taking electrons. So that's our 0.82. Okay. Our oxidation is going to be right here, negative 0.32. I could calculate my cell potential, and then I could plug it in here to calculate delta G. If you do that and you do all the appropriate conversions, you will find delta G comes out to about negative 220 kilojoules per mole, which is a lot of energy. Okay, but it turns out that our electrons don't jump straight from NADH to oxygen, but instead they're going to kind of skip down a step at a time, and each step is going to pump some protons. All right, another calculation. So calculate the standard change in free energy for this reaction. First thing you have to do is figure out what's being oxidized and reduced. Okay. It turns out that Q is being reduced. It's gaining electrons. And the FADH2 is being oxidized. It's giving away its electrons. Okay, so when we look at our table, you come back up here, we are going to look, we're going to have this value, negative 0.22 for the FAD, and Q, ubiquinone, right here, 0.1. I'm going to think, okay, it's reduction minus oxidation. I said the Q value is being reduced, our ubiquinone is being reduced, so I know that my cell potential, this would be my standard cell potential, right, would be the one being reduced minus the one being oxidized. Okay, and then you could plug that into the Gibbs free energy equation, negative NFE. All right, 2 electrons are being transferred each time, so that number is 2. Okay, and when we plug this in, 0.32 plus your Faraday's constant, you'll find this is negative 62 kilojoules per mole. When you look at this, would there be more or less energy produced when electrons are transferred from NADH to Q, or when they are transferred from FADH to Q? If we come back up here, the pink highlights were the FADH, and then here's the NADH. Notice that there is a bigger gap here than we see here, right? So we would see more energy being transferred when the electrons are going from NADH to Q. All right, back to our electron transport chain. So in our electron transport chain, the electrons are not jumping immediately from the NADH all the way down to the oxygen, but instead we're seeing this stepwise. So they're going through these various redox centers, and they're giving off a little bit of that energy at a time. So we have sequential redox reactions that carry the electrons downstream to oxygen, and along the way, energy is used to pump protons out of the matrix into the inner membrane space, setting up a proton gradient, which is the whole point of this cycle. When we look at the 4 enzyme complexes, here is a chart, some things to notice. These enzyme subunits are enormous, okay, more than 900 kilodaltons, 140, 250 kilodaltons. These things are huge. They're multi-subunit, multi-active site enzyme complexes. Okay, something else to note, notice all these prosthetic groups. Okay, this one right here, it's flavin mononucleotide. Okay, it's similar to the flavin adenine dinucleotide we looked at earlier. We have these iron sulfur clusters. So it's iron covalently bound to sulfur in the side chains of cysteine groups. So iron covalently bound to sulfurs in the side chains of histine groups. We have heme, okay, which is you're going to seem very similar from before when we talked about hemoglobin. Okay, and we have copper working to help carry these electrons. All right, you can see that some of these are on the matrix side, some in the membrane core, some on the cytoplasmic side. That's where those carriers are kind of physically located. If we look at some pictures of these, okay, you can see these iron. So the iron depends on the individual enzyme, but they could be, this one's coordinated with 4 cysteines, so the sulfurs from 4 cysteines is holding that iron in place. Here you can see the irons are held close together by various sulfurs, all right, which makes it easier for electrons to be passed down a chain. Over here, we have our heme group. It's a little different from when we talked about hemoglobin. The iron in the oxidation state here is going to go from iron 2 plus to iron 3 plus, back to iron 2 plus, and so on and so forth. It kind of jumps back and forth between those. Ubiquinone is this one way over here on the left. It's going to take an electron, okay, and we can make a couple different things. Here is our fully reduced form, QH2. So what we've seen so far is Q and QH2, the one that's gained both of those hydrogens, right, is carrying 2 extra electrons, because remember these were those hydride ions. Also, we're going to see in a little bit that we can have these intermediates where it's gained only 1, okay? Or it's gained just the electron and given off that proton. And we'll look at that in just a minute. All right, we have finally made it to complex 1. This is the complex we are going to look at in depth. The other ones we'll look at, but just not quite as in-depth. This one is really, really well understood. So let's take a look at where we are. First of all, notice this side is the matrix side. This side is the inner membrane space. Remember that our enzyme occurs along that inner membrane. Okay, so it is a, what's the word I want? But it's associated with that membrane. Okay. It is an enormous complex. It has 45 polypeptide chains. It has 14 core subunits that allow for electron transfer. It has this kind of long water channel that goes through it. Okay, it is really well understood, in part because this complex is conserved in all species. So we can isolate it from a lot of different species and made it really important to study. All right, let's look a little bit at the stoichiometry of the complex. So an NADH, okay, will essentially react in redox reaction with ubiquinone and 5 protons from the matrix, okay, and it'll convert it to the oxidized form, so NAD plus, okay, so we're oxidizing here, the reduced form of ubiquinone, so ubiquinol, and then 4 protons, but now they're out in the inner membrane space. We've pumped them out. If we count the protons, I've got 1 here and 5, so I've got 6 on the left. I've got 2 here and 4. I've got 6 on the right. All right, let's look at the first part of that. So we have this whole thing here is complex 1. We have these prosthetic groups, our iron sulfur clusters here and here and here. We've got that flavin mononucleotide here, and our NADH comes in and passes electrons to that flavin mononucleotide, and then they get passed from iron to iron to iron until they get down here, and there is this kind of enclosed chamber at the intersection of the membrane and this part of the enzyme that holds our ubiquinone. Okay, remember Q is lipid soluble, all right, and it's kind of in this little enclosed chamber here, and as it takes those electrons, it becomes negatively charged. All right, we get this Q2 minus. When that gets negatively charged, remember everything is all about the conformation, the shape, the like charges repel, opposite charges attract. So when this becomes negatively charged, it repels other negatively charged side chains, okay, in this complex. And then because of that, that conformation, it causes a conformational change. It causes movement. All right, so we see a conformational change that happens as due to this negative charge. All right, so then let's look at how that plays into pumping protons across this space. If we look at this part of our enzyme, you will see that there are some protein channels, but they only go halfway through. Protons can't just move all the way through. There are similarly protein channels on the intermembrane side that go only halfway through. The conformational change caused when Q became Q2 minus, okay, that conformational change causes changes in the pKa's of the side chains, okay, that cause which cause the H plus to bind. So remember, these are all proteins, so there's all these side chains all through here, and the conformational change changes the pKa. We saw that before in some of our studying some of our mechanisms, right? That we saw things become more acidic or basic, willing to donate or accept protons. We saw it with histidine in one particular. All right, so that essentially that conformational change allows the side chain to act as a base and accept a proton. So it will accept a proton, protons will come in and attach, and since the channel is only open to the matrix side, the protons will come in, bind to the side chains, and then they kind of get passed down to this water channel. And now they can move more laterally through here. And remember that that water channel is also open to the channels on the inner membrane side. Okay, and so those protons can hop off, attach to a side chain here, and get carried on out. Okay, so the pKa's on both sides have changed in a way that allows them to accept the protons. Remember that weak acids and bases are in constant equilibrium with water, so they're constantly attaching and detaching, accepting and giving away protons. And so it's not surprising that, oh, that proton would bind to a basic subchain here. Oh, and now it's given off, and now it's bound to that one over here, and it can work its way down. So kind of overall, this is essentially how the pump works, right? Energy from the electrons that started up here is received by Q. Q becomes negatively charged, and it will, by the way, also, think about negatively charged, going to be attractive for protons. It will eventually pick up protons and become QH2, so first it's that Q2 minus, causes the conformational change, which pull those protons from the matrix. They pass the protons to the water channel, and then they get passed off to the side chains to the inner membrane space. So the pump comes down to the fact that the charge sets up the conformational change, which changes the pKa's, which has protons attaching and detaching from those side chains. All right, the QH2 will carry on down to complex 3. So we'll take a look at that again in just a minute and see where it goes. All right, complex 2, so this one circled in pink, is also picking up electrons. We looked at this a little bit before. If you remember that FAD wasn't free to move, it was tied in to the enzyme we looked at before in the citric acid cycle. So Q would pick up the electrons, bring them in. Okay, this one is not a proton pump, but it does provide electrons and protons to complex 3. So it passes them through this uses coenzyme Q to pass them along right here to complex 3. All right, so complex 3, electrons from ubiquinol are used to reduce 2 molecules of cytochrome C in a reaction catalyzed, right, by Q cytochrome C oxidoreductase. If we look at the overall reaction, you'll see it's also a proton pump. We start with QH2 and 2 protons in the matrix, and we end up with 4 protons in the inner membrane space. So we're definitely helping set up our proton gradient. All right, let's look a little bit at how this would happen. So we have this, what they're calling Q-pool. It's just a bunch of our ubiquinone. We're going to have ubiquinol, QH2, moving into that complex 3. And cytochrome C has iron, so it's Fe2 plus to Fe3 plus. So essentially the key thing there is it can only take 1 electron at a time. If it's Fe3 plus, it can accept 1 electron to become Fe2 plus. So I should actually write this in reverse just so it's not confusing later. Let's say, as this is accepting the electron, because that's what we're looking at, going from Fe3 plus to Fe2 plus. So look at what we've got. QH2 comes in here. It gives away 2 electrons, 1 there and 1 there. But the cytochrome C can only take 1. So the other comes down and bonds to another ubiquinone. Okay, and now that one has been partially reduced. It's picked up 1 electron, and we're going to see that carried through in our next step. In this process, it's given away the electrons it was using to form those bonds, so it gives off those 2 protons. Notice that in this picture, the inner membrane space is up top, the matrix is at bottom. Now, in some of our complexes, we have this partially reduced Q. So the next time a QH2 comes in and gives one electron to cytochrome C and gives the other to that partially reduced, we get our QH2 back. So we're regenerating that. Okay, notice that we can lower the proton concentration in the matrix two ways. One is by pumping protons through out to the other side, and the other way is essentially bonding them in these in some other molecule. Both ways will increase the gradient. All right, on to complex 4. This one is not as well understood, but we're going to look at what we do know. Okay, cytochrome C has carried the electrons from complex 3 to complex 4, and now it's going to drop the electrons to a copper and a heme group. So we're going to pass these electrons off here, and they're going to get passed down and then to another iron and another copper. Okay, and once we've passed off an electron to both iron and to copper, these were originally, in the blue, the oxidized phase. So the oxidized phase would have been Fe3 plus and Cu2 plus. But now over here on the right, they're going to be in the reduced form. So this would be Fe2 plus and Cu plus. Okay, the ones shown in pink are in the reduced form. So now each has an extra electron that can be donated. Oxygen comes in. Okay, and when it comes in, it sees those extra electrons, and it essentially picks up those electrons. We get this kind of O2 minus, but it bonds and sits between that iron and copper. All right, we have essentially a peroxide bridge. Peroxide is when the two oxygens are bonded together, and so we have that. Then another cytochrome C comes in and drops off more electrons. And now that we have more electrons, they work their way down just like the last ones did, but they finally get down here to this iron and this copper. Okay, and they can eventually bond to this peroxide, and when they do that, now there's enough electrons for these things to pick up protons. So they pull those protons from the matrix, and now we get these OH groups bound to both the iron and the copper. Okay? Then, if we look at it, two more protons will enter, and when they do, now these two things will leave as water. Okay, so we made two water molecules from that oxygen. Okay. If we look at the net, we had cytochrome C coming through to drop off electrons. We saw that happen twice. We saw that we had oxygen going to form two waters. 4 protons were pulled from the matrix to be a part of the water molecules, and another 4 are pumped out using the energy that was given off. That mechanism's not really understood well. It is an area of active research. All right, that was a lot, y'all. 4 different complexes. So what should you know about the electron transport chain? We looked at lots of detail so you can understand. I don't want it to be just a black box. The overall big picture, right, is we get energy from the electrons. They cause a conformational change, which cause the pKa's to change, which allow us to move protons from one side to another. We took a really close look at details of complex 1, so you should try to tell that story yourself. You should be able to walk through that. We didn't talk as much about 2, but you should know the basic idea that it essentially takes the electrons and the protons and how it transfers them using ubiquinone. We looked at complex 3, where we looked at that ubiquinol change, transferring electrons to cytochrome C, pumping out protons. And in complex 4, we looked at the transfer of electrons through copper and iron to oxygen to pump protons out of the matrix. So you should essentially be able to tell that story for each complex to the level we covered it here. All right, why are electrons, so a couple checks, why are electrons carried by FADH2 not as energy rich as those carried by NADH? And we kind of already looked at this. And it is because FADH2 has a lower redox potential. Lower redox potential. Right, there's not as much difference between the FADH2. The NADH gives off more energy when it gives those electrons away. And the net result, after we finish looking at all of these cycles, is that less ATP will be synthesized from the oxidation of FADH2 as compared to NADH. All right, so just kind of a fun fact, that the electron transport chain is organized into a big complex called a respirosome, okay, which has 2 copies each of complex 1, complex 3, and complex 4, and they're associated all with each other, right, to make this very, what's the word I want, efficient. All right, couple questions. Which of the following enzyme complexes catalyzes reduction of oxygen to water during oxidative phosphorylation? All right, so you should study to the level where you can answer these questions without looking back, okay? So study to that level and try to answer this question. Okay, you should be able to tell the story of each of the complexes. All right. Tell you what, I'm going to come back at the very end, and highlight the answer, okay? So you can try it on your own if you want first. Which of the following carriers in the electron transport chain is a protein as opposed to a small molecule? So when I think about these, we have looked at the structures of most of these. We looked at this one today. I don't want to do that with the highlighter. Okay, this was a small molecule. This is a small molecule. We've looked at all of these structures. It's the cytochrome C. All right, following that, there are 3 questions. These are challenge problems. I want you to try them on your own. These are all info from earlier in this series and from Gen Chem, and I'll work these out in a future video, but I want you to try them on your own first. All right, I am going to go back, so you might pause the video now. I'm going to go back and highlight that one answer we skipped before I end the video, and then we will be done with this one, and you can practice and come back and see if you can get that answer. All right, stop the video.